No. Okay, everyone, can you hear me?
Yes. Yeah, that works.
Okay, can you hear me now?
Yeah.
Yes.
Okay, I keep it mute and unmute and all that. Okay. Okay, so like I say, I'd like to work on the study guide for the purpose of prepping for the exam. And as I say, the exam will be a little different. Just because I've seen some cheating and I don't know quite how extensive it is, but this time I'm going to open the test on Monday at 10, and you're going to have 3 hours to complete it. And then I'm going to open it again on Tuesday because there are some students who can't take it on Monday, and open that for 2 to 3 hours. So you'll need to complete it in one sitting. I have colleagues who've tried Respondus, which actually silences or stops your browser, but other faculty have had some trouble with it. So this is kind of the best I can come up with. And let me see. So Annette, are you there?
Yes.
Okay, I've not had this problem before um, where the people got stuck in the waiting room.
Yeah, that was me.
Well, there, I'm glad you're here. So what I want to do, I had gotten up to, I think it was about number 21 on the study guide. And permit me, I'm going to be turning to the side because I have a written copy of the study guide, and then I'm going to be putting up for you, first of all, the PowerPoint, because I want to hit some key things on the PowerPoint that are basically where I left off the last time. I would like to let you know I am recording these sessions, and when I went to YouTube to prepare them so I could put them up for you, they're long and it's taking them a lot to prep them up. So I hope to have them for you for people who've missed. But at this point, um, it's good just to be here. Before we get to the PowerPoint and the study sheet, does anyone have any questions about, now this is our last week together, really, and then next Monday and Tuesday are your final, and then the grades have to be in. But does anyone have anything you want to ask me as of now? Okay, then I'm going to go to a screen and bring up the PowerPoint. And this is, I want to give you the, wait a minute. Okay, the thing here that I want to point out, and this came from grading so far several of the antibiotic study guides that have been turned in, is that I asked you, what is the mode of action of antibacterial drugs? And I'm getting some this and that sort of different things, but keep in mind, because this is on your final, that there are a number of targets of a bacterial cell. Of course, the cell wall, protein synthesis, which we had not really hit when I met with folks last time. Cell membrane, taking care of the cell membrane, knocking it out, and then metabolic activities of the cell that are specific to bacterial cells. The whole idea between or for antibacterial drugs is selective toxicity. And I would recognize what that is, that you're trying to get the bacteria, you're trying to target the bacteria, but leave our own cells safe and sound. And then this last one down here is nucleic acid replication. And it gives you the major groups here that are involved in each one of these modes of action, or it may say target of action. Moving on, so the cell wall synthesis, these are basically beta-lactam drugs, and most familiar probably to you are all the penicillins. There are about 50 different penicillins, but they all have this in common, this beta-lactam ring. Cephalosporins also have the beta-lactam ring, and I'm just reviewing a little bit, but it won't hurt. The target is, of course, the cell wall. And remember, we have NAGs and NAMs. These are the sugars. They make up the chains. And then the connections of the chains are the peptido part, which would be the tetrapeptide to the tetrapeptide. And then sometimes there's a link of the tetrapeptide. So the penicillins all go after the peptidoglycan and the links between the NAMs and the NAMs. And what happens is the cell wall gets weakened. It can't make cell wall and refurbish it, so it's osmotically subject to lysis, and it lyses. And you see that in this picture here. I'll move on. One of the things, and you guys will be giving people injections, but just know that one of the most common injectable penicillins is G. And thankfully for many, they've developed the oral V. They have a narrow spectrum of activity, and that means, and this is something I would know, narrow spectrum means it only covers a particular group of bacteria, of organisms. So in this case, primarily gram-positive bacteria, and that's because, if you'll recall, they have a thick peptidoglycan. You can really get at that. So a few of the, again, just showing you the beta-lactam ring there, oxacillin and ampicillin, amoxicillin, very common. As a kid, you were probably given oxacillin, ampicillin, or amoxicillin, or one of those, or the injectable kind. Now, this is important too, and I do ask you this on your exam, because today so many bacteria have acquired the ability to be resistant to the penicillins. Methicillin is one famous example. So they're able to produce enzymes, which are called, as you can see here, I have in dark, penicillinases and beta-lactamases. And what those enzymes do, as you'll see here, is they target the beta-lactam ring. Once the beta-lactam ring of the drug is broken up, it no longer is effective. So it's a very sneaky way that bacteria have of defending themselves. And that's an important thing to think of this. These penicillinases and beta-lactamases are produced by the bacteria themselves. It's not intrinsic in the actual drug itself, the penicillin, whichever sort it is. And so that does lead us to methicillin. And one of the things about methicillin, notice that it acquired a plasmid. So that goes back to cell structure, all of that we had earlier, that these are tiny pieces of DNA. Very often they carry very bad information, such as antibiotic resistant, toxin production, and other things like that. So therefore these Staphylococcus acquired this plasmid, and now they are resistant. Not much use of it anymore because there is such widespread resistance. And the beta-lactam ring here, this is methicillin, and the beta-lactam ring there. And I have this here because it is so important for you guys going into the medical profession. One thing you might not know is we carry methicillin. Well, first of all, we carry Staphylococcus on our bodies as part of our normal flora. About a third of folks carry it up in their nose. And that's where the methicillin-resistant staph can hang out. So particularly if you end up working in taking care of little preemie babies and things like that, that would be very good to be tested to make sure you don't carry that. You don't have any symptoms, you're perfectly fine, but you can pass on that resistant bacteria to others. It used to be we only saw this in the hospital. So this was hospital that you picked up, or care facilities that you picked up this resistant organism, but now we see it out in the community. And so it says down here, community versus hospital acquired. And boy, we see that a lot today. Even a kid riding outside on his bicycle falls, scuffs his knee, and then ends up with a major infection. And it's not just a skin infection. This can end up going into the body, causing bacteremia, pneumonia, very, very serious. And we basically only, by the way, have a few drugs now that can take care of that. One of them is vancomycin, which we'll get to. I did put this in, just some different drugs here of the penicillin type and what they do. Cephalosporins work very similarly to penicillins, because notice they have the beta-lactam ring, little different than penicillins. And you may see when you're working in the hospital, they are designated as far as first generation, second generation. So what happened is the first generation got resistant. So then they developed the second generation and so on. I don't know how many generations of them there are now, but that is a designation for cephalosporins. Bacitracin. Bacitracin is not a beta-lactam antibiotic, but it does work on the cell wall. That's the key. And notice this, something you might want to know is it's topical. You only use it on the skin. And it was originally named for a little girl named Tracy. Tracy had a wound that produced this bacillus, and that became bacitracin. And again, this talks a little bit about its action, again, weakening the cell wall. And then comes along vancomycin. Vancomycin is a very strong drug. It can only be given, let me admit someone here. I don't know why it's doing that. I'll go back and check that. Vancomycin, as I say, is a very strong antibiotic, and it can only be given by IV. And right now, for methicillin-resistant bacteria, that is being used. And why are you stuck? Okay, let me go to your, and this is page 5 of your study sheet. I just want to hit some things here. So I had left off actually on number 21. If you happen to have been with the last little get together, but going on down 22, how are cephalosporins similar to penicillins? They have a modified beta-lactam ring. And then 23, how did staph become methicillin resistant? It has to do with the plasmid they acquired. And community-associated methicillin resistant Staph aureus is just that. You're picking it up out in the community. There have been cases of locker rooms in high schools where athletes picked it up from each other. It's pretty prevalent out there. And then down to 25, brings up a term. It says bacitracin is an antibiotic administered parenterally and affects protein synthesis. It's parenterally means usually by injection or putting it into the body. And so obviously bacitracin is topical. It's put on the skin and it doesn't affect protein synthesis. And then we get to 26, which is where we are here. Vancomycin interferes with cell wall synthesis, in particular, notice alanine to alanine cross bridges specifically. And what that's actually referring to is the tetrapeptides that come off of the NAMs. One of the amino acids on each of those tetrapeptides is alanine. And so it interferes with the alanine to alanine cross bridge that can develop for some bacteria. So going on, I can get my poor computer, come on, guy. Okay, this happens to show you pictorially what's happening. Vancomycin comes in here, settles down on the tetrapeptides, and you can't make a link. All right, then we go to antimycobacterial. We're looking at the genus Mycobacterium, which number 27 is referring to. In particular, we're back at the cell wall, but something very specific. Remember that waxy coating that the Mycobacterium have, this mycolic acid. It interferes with its incorporation into the cell wall. Mycobacterium have peptidoglycan, but this is really key, really to their pathogenicity, their cell wall structure. So when they lose that mycolic acid, they can't survive. So a few drugs here, isoniazid, used to on Mycobacterium tuberculosis, ethambutol, and so I said, name an antimycobacterial drug. One of those would be just great. And they're still used today. Treating tuberculosis is very difficult today. It takes a whole group of antibiotics, and then what happens is they have to alternate them. A lot of these drugs make people nauseated, and they don't really want to take them. Okay, what else? Okay, going on. So now protein synthesis, and we go to number 28 on your study guide, and this is something I asked you on the exam. One of the things that I did on your exam is I wanted to probe a little bit of how much biology you have acquired up to this point. And one important thing is the ribosomes. What do they do? Yes, it's protein synthesis, but why is that really important for a cell? A cell cannot survive without protein. You know that the cell wall, for example, for microbes, well, for bacteria, for us, this is really key. We cannot live without enzymes. So enzymes in metabolism, very important. Channels and pumps in our cell membranes that allow certain things in, restrict some things to get in. So I do ask you about this, just to make sure you know why proteins are so important for any organism. And then this leads to number 29, which is, what is the major difference between the bacterial ribosome and eukaryotic? Well, it's right here. I've noticed on the study guides I've graded, they're kind of missing out on this. The 70S, S remember stands for Stedberg unit. Has to do with the size and when it drops out during centrifugation is smaller. So the 70S, and what in the world did I do there? So the 70S ribosome is for prokaryotes. What's the eukaryote? Well, the eukaryote is 80S, and so there's a difference in the size of the ribosomes. And so we can target bacterial ribosomes and leave our own ribosomes alone. This was just taken from what I think your textbook, yeah, Pearson, just showing some different ways in which we can interfere with protein synthesis. Coming down to number 30, before we go to chloramphenicol, I just want to point out something. It says, what part of a eukaryotic cell could be affected by antibiotics that target ribosomes, and why? Anyone have an idea of where I'm going there?
Mitochondria.
Right, exactly. Very good. Ivan, was that you?
Yeah.
So exactly. So mitochondria and of course chloroplasts, but we don't have chloroplasts. Mitochondria have 70S ribosomes. And that goes back to some evolutionary thought about the development, remember, of the eukaryotic cell coming back from bacteria, that the mitochondria may have originally been bacteria that got incorporated into a cell. They took up a relationship, a symbiotic relationship. But those mitochondria are really cool. They have 70S ribosomes. They have their own DNA that looks a lot like, well, more like a plasmid, really. And they have enzymes of their own that they can make. So that is the answer. I do ask that on your exam. That's one of your questions. And so we go to, actually, I may be a bit off here. This is chloramphenicol. This used to be used it targets the 50S subunit. I was talking with a member of one of your guys' classes who works in the pharmacy at the hospital. And this is so ghastly, they don't really use it anymore. Suppression of bone marrow activity, all kinds of things. They've phased out using chloramphenicol. And so we are in tune with the study guide now, aminoglycosides. And that brings us to, well, a couple of things. These are pretty common, streptomycin, tobramycin, neomycin, and gentamicin. Neomycin usually incorporated as a triple antibiotic for taking care of wound infections. But notice one of the first that really helped gram-negative, because the penicillins pretty much take care of gram-positives, and it works very well with other drugs, acts synergistically with other drugs. And so we go to number 32, aminoglycosides. Then it says can interfere with what? Your answer should be what for number 32, if you happen to have it.
C.
Yep, C, exactly, the 30S. Okay, and then it goes on again. Why might a physician hesitate to prescribe chloramphenicol or an aminoglycoside? Notice permanent damage to the auditory nerve. I'm running out of time. Damage to the kidneys, etc. So have to be careful with these drugs. Notice neomycin, primarily topical. I'll give us about 5 more minutes, and then you can join me this afternoon or tomorrow morning. If you guys want additional get-togethers, I'm happy to do that. Tetracycline, what are some things that we should know about tetracycline? If you'll come on down, a common antibiotic added to animal feed. It tends to buff them up. They gain weight better, but also if you notice today, animals are kept very much in feeding areas where the animals are all put together. You know, it's all kind of almost cruel to me to see, but they're more likely to pick up infection from each other. So tetracycline used for weight gain, but also to protect these animals against infection because they're so packed together. That is what it looks like. It looks like a really nasty fungus, but it's kind of part fungus and part bacterial in nature. I just had to show you what that looks like. And very importantly, when you get into giving prescriptions out, notice it's not a good thing for children. Causes brownish discoloration of the teeth. So as the adult teeth come in, they're discolored. And I had a student who had that happen to her. Her teeth were perfect, except they were very discolored. And there you see the essence of what it looks like. Okay, I will stop. Hopefully, if I don't go too far, I will be able to retrieve this better for to do, you know, a little, take the recording. So my plan is to this afternoon at 1:30 to continue this, and then tomorrow at 10 o'clock to actually hit the final and to really talk to you more directly about exactly what you should study. You can have, here's the deal, just like last time, you can have your study sheets here, you can have your PowerPoints, your book. The only thing I really ask is you not Google, because to be honest, I can tell it's doesn't, the answers are not microbiological ease. They don't sound right. So try and stick with the goodies you have, because really the things I ask you are right here in your handouts. So I'll be more explicit tomorrow with you in terms, and this afternoon as we wrap up this antibiotic study guide. Before I cut us off, anybody, any questions, thoughts, anything you want to share? Okay, well, thank you for coming, and I hope to see you next time.